guess, I'm sure. So it should be started. I'm yellow, I'm green. I hear a distinct uptick in my fans.
We're on.
We're on?
We are on.
Hi, everybody. How's it going? Yeah. You want to introduce yourself, Bob?
Yeah. I'm Bob Kuhn. I am teaching for the last almost 20 years at Centennial High School in Roswell, Georgia. And I'm happy to spend my Friday evening helping out some folks with review.
Yeah.
Also do the moderator on the teacher community. So I guess both of us do have or have had some connection with College Board in a way. So we
Definitely
have a little bit of insight to this.
Sure, but we should also probably clarify. First of all, I'm Dave Knufke. I used to teach this subject, but recently I have stepped away from teaching it directly, but it's definitely something I know a lot about. And Bob had hinted that, we have had positions for the College Board, but never in a test development capacity. I've never attended a reading. Did you go last year, Bob?
No, actually, this summer will be my first for bio.
Yeah, so keep that in mind if you're watching this. We are just two teachers more than anything else who have done this quite a bit of time for students. There should be some moderators over on the side, so if you have any questions, you can hook them up. I think I'm asking a lot of my computer right now, so the feed may go in and out. You know how it is on a live stream, so you guys will have to bear with us. So the way that this is going to work is we're going to go over the 2013 released sample exam. So this is an exam that is publicly available on the College Board website, and there's a link right down below. If you don't have the exam in front of you, this is not going to work well for you. We will not be projecting anything from the exam. We will not be showing images from the exam or anything like that. So you really kind of need to have it out in front of you as we go through it. Otherwise, this is going to seem like a very strange kind of session, I would guess. So make sure that you get the exam. If you don't have it yet, you can download the PDF, I guess, and follow along. You probably want some paper. You probably want a 4-function scientific calculator, maybe, so for any calculations, because that's the kind of calculator you're supposed to have. There's Bob's. That's the kind that my students use as well. I seem to have lost my own one. I think my child has taken it and has used it as a fake telephone, which is better than him using it as a real telephone. But, right now, but I do have a scientific calculator, but I'm not going to hold that up because you can't bring a scientific calculator to the exam. And so since you can't do that, you don't need to see that here, certainly. So we'll forgive you that one. The other thing is that we are both teachers, as I said before, and as teachers, pardon me 1 second, little window problem here. As teachers, you should understand that we're going to look at this exam a little bit differently probably than you will, right? There's the difference between sort of beginner's mind and the expert mind. And I mean, it probably sounds really fancy. I don't mean it to sound this way, but Bob and I are quote-unquote experts, just meaning that we've stared at these things a long time. And so when we look at these questions and talk about how to do them, it's going to be fundamentally different than somebody who's only been doing this for less than a year. So please don't feel bad if we go through questions really quickly and you're like, how do these dudes do that? I can't do it like that. That's kind of the point, right? Even though, beginners and experts approach exams differently, there's value as a beginner sitting out there on a Friday night or whenever it is you might watch this in watching how experts go about sort of thinking their way through a test like this. And hopefully that will be useful for you. And if it's not useful for you, no harm, no foul. This is, the cost of this session is free $99. So as a result, it shouldn't be any big deal. You got anything else you want to say, Bob, before we tuck in?
No, I guess we're going to spend a minute or two on the exam structure, and then we'll get right into it.
Yeah, so the exam, is split into 2 parts. You've got part 1 and part 2, and that's really what you're going to see. And looking at the practice exam, and I have mine in front of me here, the front of the document really is just the verbatim copy of the instructions that the proctors will read. These might change a little bit from time to time, but they're not going to change that much. So you could look through that if you want. There's definitely a thing about what you should bring for the exam and what you should have while you test, right? You probably want some pencils. You probably want some pens. Depending upon your district, or your school, either a four-function calculator will be given to you or not. They give you a copy of the actual answer sheet that you will actually be filling out, which is actually useful, particularly for the grid-in questions, right? The grid-in questions are on page 3 of the answer sheet, and you can see what those look like. It's definitely a format that you want to be familiar with before you walk into the exam so that you know how to fill them out. So you can see them right there on the middle of page 3. Yeah, and I mean, that's about the front of the exam. I don't know, Bob, if you have anything else you want to say about the front of the exam. I'm putting my copy in the garbage.
No, just if you're a first-time exam taker, don't let the fact that the grid-ins go from 121 to 126 throw you.
It's a good point.
Exam's not that long.
Yeah, absolutely. The other thing that we'll point out here is that the exam that you have is a released sample exam, and so as a result, 10 multiple choice questions have been removed from it, and 1 grid-in question has been removed from it. This is for fancy College Board purposes. There are some number of items that are put on the exam for internal purposes. These are not really figured into the overall score, but we don't know what those are, and you won't know what they are. They're not like the last 10 or the first 10. So as a result, you have to treat every question that you see as if it is a question that will be scored. But you can see when you get to the actual exam, right, when we get to part 1, the multiple choice questions, after looking at the equations and formula sheet and the instructions, you can see that it says 53 multiple choice and 5 grid-in. And that's different, right, because we only have, your actual exam will have 63 multiple choice and 6 grid-in questions.
Yeah, and I don't remember if you said this, David, but the exam you're seeing is not the exam that kids took in the U.S. This is the international exam that was given. And so, the teachers have had this exam for a while, secure document, but this is the first time that students have seen this fresh as a released exam. So it's really a great tool, I think, to release it.
Absolutely. Super, super useful. So with that in mind, let's talk a little bit about section 1 of the exam. And you can see the instructions over on, I think it's, the numbers in this document are a little bit discontinuous. So we get to section 1 multiple choice, and then they start numbering the pages over again. So we have on page 2, they give you the instructions. It tells you how long you're going to have. It reminds you how to fill in a multiple choice question. You're not going to be asked about Chicago on the exam, but is there for you as a demonstration. Tells you how much time you have, what the percent of the score is going to be, what you need in order to take it, and you can see all of that on the document. And then you get the formulas, right? You get the equation and formula sheet. And so this is your friend. This is definitely something that you want to just probably keep a handle on the test, because you might need to use it to answer questions, and as a result, you should be able to pull it out. The rule of thumb, if you have 63 questions and you have, the 1 hour and 30 minutes in which to do it, if you try to budget yourself to 1 minute a question, you've got 63 questions and 6 grid-ins. That's going to give you some time at the end to go back to questions that took you longer. So that's kind of a good rule of thumb to just keep in your head. If the question is taking you considerably longer than a minute to answer it, you might want to just tag it, some symbol on the test, right? Just maybe dot the question, and then move on and come back later, because time is of the essence, as you probably have found out if you've taken any practice exams. So before we tuck in to the exam, the only other thing that I have is I have a list of shout-outs left by teachers out there for students out there, and so over the course of today, we'll be going through some of these shout-outs. First shout-out comes from Chris Robson, who would like to send a shout-out to Robson's AP Bio classes over at Air Academy High School. Mr. Robson says that his classes, or Ms. Robson, Chris is spelled ambiguously, and I apologize, Chris. The teacher, known as Chris Robson, say, my classes have that yay biology spirit, and so if in fact they do, then we know that many of them are out there watching this right now, and I've angered all of them with my confusion over the gender of the teacher known as Chris Robson, but I apologize. So let's tuck in. We'll start with question number 1. So you want to do it, Bob? You want me to do it? What do you think?
I can start, I guess.
Go for it.
So I guess I don't need to read the question. It's in front of us. So I think for me, when I look at this question, we've got describing how the dog's brain integrates the information for smell.
Yeah.
So, the first thing you should always do is understand what the question's asking. So, when you go through these answers, we've got some obvious ones that aren't going to work. So, chemoreceptors in the brain, that's probably not where they're going to be located. So the brain is sending impulses to the nasal cavity, that kind of backwards.
Right. It's exactly backwards.
What's that?
It's exactly backwards. You're absolutely right.
And then for C, the tongue is kind of a distractor because we're looking at smell. And then for D, they throw in action potentials, which don't really have anything to do with this question. So the correct answer for me would be B, because we've got chemoreceptors located in the right place, which are in the nasal cavity, and they're sending impulses to the brain. So that's how I would approach it.
Yeah, I mean, I think it's exactly how I would go about it too. When I read a question like this, sometimes I might try to think in my head about what we're talking about here. And it's totally okay during the exam, if you're reading something like chemoreceptors in the nasal cavity, you can kind of just, touch your nose and think about it for a second and think, is that how this is working here, to get a handle on it? But definitely use your process of elimination as well, right? Chemoreceptors in the brain, we know the brain doesn't receive anything except incoming nervous information from other receptors in the body. So we can get rid of that off of the bat, right? Because that is just not a thing that is. Chemoreceptors on epithelial cells of the tongue. Well, the tongue's great, right? But the question here is the scent of a jackrabbit. So no, it's not going to work for us. And then receptors originating in the nose send action potentials to the motor regions of the brain. I don't know that would necessarily work here for integration. So as a result, the best answer here is, I would agree, is definitely B.
Yep, and that's the correct answer.
Cool, man.
One for one.
Looking at number 2, thrips, right? We get a lot of information about thrips. They feed on rose pollen. The population increases in the spring and decreases during the summer. The researchers hypothesize that food abundance is the limiting factor for the population. So what do they want? They want to collect data in order to figure out if that's the case. What would we measure in order to measure food abundance? Amount of sunlight is right out, right? You're not going to get a good analog for food abundance by measuring amount of sunlight. Temperature is gone, right? Because we're interested in food abundance, right? Temperature doesn't tell us anything directly about food abundance. There's probably a better answer out there. So then we have 2 things about pollen. And that's a good multiple choice tip. A lot of times if you have 2 answers that are kind of about the same thing, usually that might help you zero in on the correct answer if you're totally lost. So is it density of rose pollen produced in grams per square meter, or is it amount of pollen produced by each flower? Every time that I gave this question, the most popular wrong answer was D, right? The amount of pollen produced by each flower. And the correct answer here is C. And so the thinking here is, why is that better? Well, if you think about plants and the amount of pollen that they produce, and that's what the thrips feed on, right? The amount of pollen is going to be variable from plant to plant. And so if we want to get a handle on the overall abundance of pollen in the environment, it's probably a better idea to get a measurement of the density of pollen per square meter in the environment, so that we know we can get rid of that variability from plant to plant and just look at the total amount of pollen that's available in the environment by boiling it down to that per square meter kind of approach.
Yeah, and I didn't think of it that way. I focused in on most useful and regular intervals, and I thought scientifically it'd be much easier to sample density over time than going around to each individual flower. And
yeah.
So
that's another really good. I've never thought about it that way before. That's a perfect way of going about it too, right? The experimentally just going around from flower to flower is going to be an interesting experience. Definitely. Bob, you want to do number 3?
Sure. So we've got some ATP breakdown here, and it's going to be inhibited. So one of the things the College Board really likes to do is break stuff.
Yeah.
Have you think about what happens once it's broken. So what we're looking at here is what's inhibited in terms of movement across cell membranes. So you know that oxygen is going to diffuse, so oxygen is probably not going to be affected by this. Plus, there's no use of ATP in transporting oxygen. Same thing with water. That's a passive transport through these aquaporin proteins. And then for D, facilitated diffusion, but it says permeable substance. So for me, the most logical thing to know is that active transport transports things against a gradient, and you need energy to do that.
Yeah.
So that would be the one that's affected the most.
Yeah, I see this a lot on AP questions too. It's a sort of like one of these things is not like the others. And if one choice is very different from the other ones, it's probably a good inkling that that might be your correct choice. Of course, what you need to be able to do is understand how the things are similar or different from each other. And so if you look at these, right, choices A, B, and D are all passive transport. And C is active transport, right? We're going against the concentration gradient, and that's the only thing that requires energy. So that's the correct answer. Nice.
Yep.
Looking at number 4, if we look at the graph, we have a population that's relatively stable, right? It's cycling a little bit, and then we get this landslide event, and all of a sudden the number of individuals in the population decreases dramatically. You may remember from evolution that we call that kind of thing a bottleneck, right? And so we're going to expect some sort of bottleneck effect here that should clue us into thinking about genetic drift and those kinds of thoughts before we ever look at the prompt. If we look at the prompt here, we've got undersea landslides and some things that happen. We see the size of a population that lives on the ocean floor affected by the recent landslide. We've greatly reduced the population. So which of the following best predicts how the population will be affected by the landslide? Surviving organisms will evolve into a new species? Man, can't predict that kind of thing at all. That's just not a predictable thing. Might happen, might not happen, but I would think long and hard before going with an answer that reads like that on any sort of AP exam. The reduced population will likely have allelic frequencies that are different from the initial population. Well, this is a genetic drift situation, and we know that in the genetic drift, one of the classic hallmarks of genetic drift is that allele frequencies are different afterwards than before. So that one I definitely can't rule out. The population will adapt to deeper waters to avoid future landslides. That's essentially the same thing as A, right? We're making a statement about the future evolution of this population, and that is not something that science really enables us to do with any certainty. We could make, right, some sort of predictions if we had a rich data set to do that. We don't have that here. All we have is the number of individuals. And the reduced population will have a greater number of different genes than the initial population. If we have fewer individuals, I don't know how we could have a greater number of different genes than when we had more individuals in the population. If you want to do the math on that and show me how that works, I'd love to see it. But to me, that doesn't make mathematical sense. So the correct answer here has to be B by my reasoning.
And the other thing I didn't like about some of the other answers is this finality of will,
Yes.
will.
Yes.
And then, the correct answer has will likely. So, that kind of bothered me too as I was going through and picking my answers.
Yeah, those kind of statements are always going to get us into trouble, I think, in science. So as we move to page 6 is, of course, brought to us by Clifford Nefrata, who would like to extend his great thanks out to the folks at Arcadia High School in Phoenix, Arizona, the Chaos Corners, AP Bio Titans, and Mr. Nefrata would like to say, may the PDAs, the FAs, the CMs, and the epicness that was the 12-week theme for CM forever haunt your dead of nights. Go forth and remember, apoptosis is programmed cell death, and the mitochondrion, singular mitochondria plural, is the powerhouse of the cell. Controversial statement there by Mr. Nefrata, but we'll let it slide here on Cinco de Mayo. So let's take a look at question number 5. Bob, you want to work us through it?
Yeah, so this is looking at a most relevant statement. So in terms of the Calvin cycle. So, some of these answers might be appropriate, but we're looking for the most relevant answer. And so there are a number of distractors here that are just wrong. So for A, Calvin cycle, we're not capturing light in the Calvin cycle. Right. We're not reducing NADP plus to NADPH in the Calvin cycle.
Both light reactions.
Yep, and we're not producing ATP either. So all of those are light reaction events. So the only thing left is how's ATP used in the formation of 3-carbon carbohydrates. And, if you know that G3P is one of the outputs, you could get it that way too. But
yeah, I mean, the purpose...
elimination, you can do it.
It seems to me the purpose of this question is to take students who have a muddy understanding of photosynthesis and separate them out from people who have a very clear understanding of photosynthesis. They're not asking you about specific, steps in the cycle or anything like that, right? They're asking you about a fundamental question about the Calvin cycle. And the other 3 questions they give you are all pretty fundamental questions about the light reactions, which is why, right? B is definitely the most relevant question. My students have always had problems with these, like, pick the most relevant question ones. It's usually trickier when there's a diagram on it, and maybe we'll see some of those a little bit later on.
Right.
Cool. Number 6, Rosalind Franklin. So we know we're talking about the determination of the structure of DNA. And so the X-ray diffraction images, which of course, right, Franklin's X-ray diffraction images were in an X shape, which we know was sort of a crucial piece of information for determining that the shape of the molecule was helical. Most directly support which of the following claims about DNA? The ratio of base pairs are constant. That's a weird phrasing for that statement. I mean, the ratio of base pairs are constant in every cell in my body, right? They're relatively constant if we compare every cell in my body with every cell in every other human's body, not counting, like, you know, haploid cells. But I don't think they're constant if we compare, like, me to a bacterium. So I don't know if I love that. The nucleotide sequence determines genetic information. That's a true statement, right? But I don't know how the X-ray diffraction pattern really tells us anything about that. The 2 strands of DNA are anti-parallel. That's a true statement, but I don't know how the X-ray diffraction pattern tells us anything about that either. The basic molecular structure is a helix, right? So that's definitely the answer to this question. And, you know, that's the kind of thing that you know or you don't, really. There's not a lot of those anymore on the AP exam, but this is one where you just, I mean, you got to know Franklin's X-ray pattern led to an understanding that DNA was helical in structure.
Yeah, that would be a real hard one if you didn't know that.
Basically impossible, right?
Yeah.
Now, we should probably talk for a second about that. Like, that doesn't mean that you, if you don't have that committed to memory right now, that you should go and commit that to memory. I mean, it's probably a good idea if you want to be a biologist to kind of have that in your head. But, you know, you could memorize, oh, Franklin's X-ray pattern, the X shape indicated a helical structure, and it may not show up on Monday's exam, right? It just because it's here doesn't mean that's going to be on another exam. You know, what you get with these questions is a sampling of sort of the ground level knowledge that you need to bring into the exam, and they're going to pull from certain areas.
Yeah, and I think, you know, this being the first year, I guess, after the redo of the course,
yeah,
I think the
exam
point was still evolving at this point. And like if you look at some of the exams since then,
fewer of these.
Different.
Yeah.
So,
yeah, and I mean, of course, the exam is still evolving, certainly. Bob, you want to take us through number 7?
Yeah, actually, I emailed you about this one a long time ago. I don't know if you remember that, but so you have a formula up there. You've got a hydrogen proton, and then what is that?
It's a carbonate ion.
Yeah, carbonate, and then water and carbon dioxide. And so you've got an athlete who stopped exercising. You've got a blood pH that's dropped below the normal level. And you have to think about how you're going to restore the normal pH. So you've got to think that something's going on here with protons.
Mm-hmm.
And so some of these things don't make sense in light of that. So an increase in oxygen concentration in the plasma will lead to an increase in protons. Well, if you're increasing the protons, you're going to
decrease the pH. If you're increasing the protons, right, you're going to decrease the pH even further. That's not going to work for us.
Yeah, I mean, decrease the pH,
yeah,
right.
No worries.
We're acidic. So, and then same thing for B, temperature, I don't know where that factors, but you still get an increase in the proton concentration.
Correct.
Sweating is going to lead to a decrease in both hydroxide ions and protons. I don't know if you're doing that to both of them. I don't know how you restore the blood pH that way.
Correct.
So then in D, you're going to decrease the number of protons in concentration, and that's going to bring it back up to normal pH. So
yeah.
I would say D.
So this question seems to be throwing a lot of chemistry at us at the top, right? It's giving us an equation in equilibrium. Now, I think that it's fair kind of chemistry to assume that students probably should have a handle on. Most students would have a handle on before they sat down for the AP exam. But at the same time, like my inclination, because I taught a lot of chemistry, was to work it out Le Chatelier wise. But then once I looked at the options, you can see that these options aren't even really dealing with that. It's just saying that the athlete's blood pH has dropped below the normal level, so it's gotten more acidic, right? We know if pH goes down, the concentration of protons goes up, and I believe that that is given to us on the formula sheet in disguise. If we go to pH calculations. I'm not pulling it out right now, but I think right down at the bottom on page 4, pH is equal to negative log of the proton concentration, which is a fancy way of saying that if pH goes up, right, the concentration of protons is going down. That's the mathematics that are involved there. So pH is here, pH is going down, which means the concentration of protons is going up. So if we look at these, right, an increase in A and B, both talking about increases in proton concentration. So that's not going to work for us. That's going to bring our pH further down. And C, as Bob pointed out, if we lose protons through our sweat, but we're also, for everyone we're losing, also losing a hydroxide ion, that's going to have no net effect on the pH. So we need the thing that decreases the proton concentration. And that's given to us in the answer in D. And that's the only one that talks about a decrease in proton concentration, which is why it's the correct answer. Nice. Hey, Bob, is page 7 brought to us by anybody?
Oh, hey, it is. Let me get my shout out here. Kim Dimitro, PMSA Pythons, the Proviso Math and Science Academy.
I would have said Proviso, but it's one of those two, right? Okay. We're right. Yeah, either way, we're right.
Zoe thinks that all the AP Bio students will rock their exams, and I am agreeing.
I have no data that suggests that Zoe is ever wrong in her thinking, so it all works out. Nice. So let's look at page number 7. We're looking at number 8 here. A researcher is investigating the relationship between the existing species diversity in a community and the ability of an introduced non-native species to destabilize the community. Which of the following graphs most consistent with the claim that communities with high diversity are more resistant to change than are communities with low diversity? Well, if that was the case, we would expect that if diversity was high, the invaders would not survive. And if diversity was low, the invaders would survive. We've got survival of invaders on our y-axis and diversity on our x-axis. So we're looking for something that slopes downward as we go across our x-axis from left to right. It is D. And at that point, I would circle that and move on.
Yep.
Bob, you want to do number 9?
Yeah, actually, I understand this experiment, but these answer choices kind of gave me a little trouble. So we're looking at the famous Avery, McLeod, and McCarty transformation experiment. So you have to know a little bit about that experiment. I think it's probably one of the 4 early experiment ones I would know. Messelson, Stahl, Avery, what else?
You got your Measles and Stahl, you've got your Avery, McCarty, and McLeod, you've got your Chargaff data, right? Yeah, and Chargaff. You have your your Hershey Chase.
Hershey Chase, right? Yeah. So, I mean, you have to think about the transformation experiment. If you remember that, this is a hard one if you don't remember what that experiment was about. So, here we have a number of enzymes that seem like they would make sense. So the proteases and the NRAases to rule out protein and RNA as transforming factors. That makes sense in what we know about protein synthesis. So we'll keep that one on the table for now. Lipase is a legit enzyme. It's going to break down lipids. So ruling out lipoproteins as transforming factors, well, what we know about that today, that doesn't really factor into that process. So right. For me, I would exclude that one. We know that kinases are important as well as enzymes, but really we know now today that the phosphorylation is not happening in protein synthesis. And then ATPases to show that it's not dependent on ATP. Again, I mean, I know that ATP is used during that process, but, I think I narrowed it down to to A and D, but I sort of knew, as a teacher, I sort of knew what the answer was. But as a student, I was thinking that's where I would go, and I'd have to try to kind of choose from there. So
yeah, I mean, you'd have to know your experiments, right? And you have to know what they did in this experiment, right? So they used purified protein, and they used purified DNA, and they used purified RNA in order to determine which sample was the genetic material in bacteria, right? And was able to transmit the virulence factor. So if I have how do I purify a sample of DNA, right? I have to use proteases and RNases in order to remove the proteases and the RNases, right? How do I purify the other ones? I got to use those combination ones in there in order to get that all taken care of. And so as a result, right, A is the closest to the details of the experiment, and so as a result, that's the one I go with.
Right, and I think knowing the experiment, that makes sense. If you don't know the experiment, this is going to be a really hard one for you to narrow down.
Yeah.
Just because.
If memory serves, and I could be wrong here, and I'm sure a moderator will correct me, but if memory serves, that experiment is not an illustrative example in the curriculum module, in the curriculum framework. That experiment is actually in the it's an essential. It's up a level. It's not something that really I think instructors are given an option to teach. It's really something that teachers are really told explicitly they need to know this.
Right.
Yeah. Cool. Let's look at 10 through 13. So here we have a series. I'll just kind of work through the whole series, I guess. So the figures below show changes in populations of 2 species of flower beetles, and they give us names for the flower beetles. In cultures with parasites and without parasites, each data point represents the mean population size from 10 cultures of dishes of equal size and food content. So it looks like if we have both populations, it looks like with or without the parasite, Tribolium confusum doesn't really seem to be affected by the presence or absence of the parasite. Whereas Tribolium castaneum over time does seem to be more affected without the parasite or with the parasite, right? Am I interpreting this correct, Bob?
Yeah.
Cool. So under which of the following conditions is the observed number of beetles per culture dish the greatest? So they give us different options. This question is just determining whether or not we can read a graph, right? We see that castaneum will have higher numbers than confusum. And so if I can find a point in my data that corresponds to when castaneum is at its highest, that's going to be the one that I'm looking for. If we look at castaneum at 100 days with the parasite, that's pretty low. So it's not super low, but it seems kind of similar to other ones in the confusum. If we look at castaneum with the parasite at 600 days, that's pretty low as well. So man, now I got to go in and do a little bit more comparison here. T confusum with the parasite at 500 days, that does not look as low to me as T castaneum with the parasite at 100 days. I'm sorry, that looks lower to me. Yes? Yeah. And then T confusum without the parasite at 300 days. 300 days, T confusum, it still looks like we're below that T castaneum with the parasite at 100 days. I believe that is the correct answer. Am I right? Am I right there, Bob?
You got it.
Yep, nice.
C is the right answer.
Yep, just pulling off my points, right? C is the correct answer here, and just rocking through those graphs as quick as possible. Yep. The data over the duration of the experiment provides strongest support for which of the following conclusions regarding the effect of the parasite on Tribolium populations. This is question number 11. Confusum is adversely affected by the parasite while castaneum is not. Confusum doesn't really seem to have any difference. So I don't think it's really affected by the parasite, whereas castaneum seems like it's really affected by the parasite. So I think that's exactly backwards. Castaneum is adversely affected by the parasite while T confusum is not. That is got to be the correct answer. And so I'm going to go with that one. Yeah. Now be careful here because you might be inclined to pick C. Both of them are adversely affected by the parasite. But if you look at the data for Tribolium confusum over time with and without the parasite goes back and forth. It's not very clear at all that it's affected. And if you look at the end over at 900 days, it looks like there's slightly more, though I don't know how significant that difference is, of parasites of beetles with the parasite than without. So that doesn't work for us here. And certainly they don't both show increased fitness in the presence of the parasite. We can rule that one right out. So definitely B is the correct answer here. And that's, I think, playing a little bit upon naive misconceptions of what a parasite is and how a parasite works, right? A lot of students will see parasite and just be like, of course it has to be negatively affected by the parasite. But apparently it's not, based on the data. In Figure 1, the difference between the two curves can best be attributed to which of the following. So in Figure 1, it doesn't look like there's a whole lot of difference between the two curves. So difference between the laboratory conditions and natural environment, they're not in two different environments, so that doesn't work for us. The effect of the host on its parasite, I don't have any real information to help me pick that one as a correct answer. The influence of competition for limited resources, I don't really have any information about competition here, so I wouldn't pick that either. The natural variation among populations, that does seem to account, as best I can figure, to this little fluctuation between the two populations. That's the one that makes the most sense. This is a classic AP strategy that I see on questions all the time, where they're giving you things that make intuitive sense, but they're not giving you information about them, right? And you need to not conclude things outside of the domain of the information that you have access to in the question. I see that all the time with students. Then looking at number 13, if the experiment was continued for an additional 500 days, the population density of Castanium with the parasite would most likely stabilize at a value closest to which of the following? Well, it looks like we're pretty stable from about day 300 onward, and it looks like we're, with the parasite in Castanium, it looks like we're somewhere around 10 beetles per dish. So I'm going to go with 10 beetles per dish.
You got it.
Boom! And now we're on to a different series. Or just a different question. Rob, you want to take it?
Yeah, so we've got whales that are pooping at the ocean surface, so that's always a fun thing.
Yep.
So they're depositing feces at the surface, and that's going to be nutrients for algae to bloom at the surface, and that's going to ripple down to the food webs and food chain. So the question here is, which of the following best predicts what would happen if the whale population decreased? So we're taking the whale out of the scenario. So for A, there would be a reduction in surface nitrogen concentration, which would be true, which would cause an algal bloom.
Backwards.
That doesn't make sense. That
Yep.
would be the opposite. So we can get rid of that one. The surface fish population would decline due to reduced populations of algae. That makes sense because you don't have the nutrients anymore. The algae are not going to bloom, so fish would decline. So that would be my choice.
Yep.
If we look at the last two, accumulating mutations just doesn't even make sense. So I don't know why that would happen. And then D, the remaining whales would be forced to forage in the deepest parts of the ocean. Maybe, but
who knows?
It doesn't have anything to do with the question asked.
Who knows?
Yeah.
Yeah.
So B is the most is the answer I would choose.
Definitely. Looking at 15, so we have three different things here, crossing over, conjugation, and fertilization. And man, so now they're asking me, what are these? These are all similar in some way. So I got to know enough to know that in crossing over, homologous chromosomes exchange genetic material, and in conjugation, bacterial cells exchange genetic material, and in fertilization, of course, sperm and egg exchange genetic material. And as long as I know those things, I think I can probably figure my way through these. None of those are transcription, right? Transcription is something fundamentally different in our understanding of genetics. An increase in genetic variation, well, an exchange of genetic material is going to increase genetic variation. So that seems like that's the correct answer. An increase in chromosome number, I mean, that's true in fertilization. It's not true in the other two. And then horizontal gene transfer is, of course, the thing that happens in conjugation, but it doesn't happen in the other ones, right? That's a bacterial process for the exchange of genetic material. So yeah, it's B, right? I don't have enough time to dwell on this test, so I'm going to move on from there. But before I do, I'd like to let you know that the next series of questions are brought to you by the students of Ms. Minu Basu, the Basuologists over at Doherty Valley High School. And Ms. Basu would like to wish good luck to my awesome students and everyone watching this session. So take it for what it's worth, guys, because it's worth a lot. Robert, do you want to do number 16?
Sure. So we have a developmental question here. So We have a vertebrate forelimb that's developing as a solid mass of tissue, and then as it starts to develop even more, it's getting modeled into individual digits. And so this question's asking, which of the following explains the role of apoptosis in forming this forelimb? So in this question, if we take them one by one, the apoptosis isn't going to replace old cells with new cells.
It's not what it is.
Is getting rid of cells, so we can cross that one out. Apoptosis involves the regulated activation of proteins in specific cells of the developmental forelimb that leads to the death of those cells. So to me, this is going to be the one I'm gonna think about, because this is pre-programmed cell death that's dealing with some sort of activation of genes. So this is going to make sense to me.
Yep.
For C, the destruction of extra cells, which provides nutrients for phagocytic cells. That doesn't make any sense to me at all. In development, nothing I know about development. And then the last one, could be kind of tricky. Apoptosis in developing forelimb triggers the differentiation of cells whose fate was not already determined. That sounds real smart, but if you think about the way this works, it's actually the opposite, right? The fate is determining the apoptosis or the gene expression, so it's kind of a backwards statement. So
yeah.
I would definitely go for B.
Yeah, I think this question also is a good example of a thing that you see on the AP exam sometimes, where they basically are asking you, what is the definition of apoptosis? And they have given you a bunch of other stuff here that kind of hides the fact that they're asking you, what is the definition of apoptosis? But if I asked you on a question, if I removed that entire blurb up top and I just called it, what is the definition of apoptosis, right? B would also be the correct answer for that, right? You see that sometimes as well. If you can recognize those, that helps to save you some time digging through those stems. So let's take a look at number 17 here. So we have a graph. The oxygen concentration goes up during the day. It goes down during the night, during the hours of darkness. Light seems to be leading to an increase in the oxygen concentration. I know that oxygen is produced as a product of photosynthesis, and photosynthesis requires light. So what most likely causes the trends in oxygen concentration shown in the graph above? The water becomes colder at night and thus holds more oxygen. I don't know. I mean, yeah, colder liquids tend to hold more dissolved gases, but that doesn't, but it's going down here, right? Yeah. So that doesn't seem to make sense. Respiration in most organisms increases at night. I don't know, man. I don't know. I would argue that when organisms go to sleep, right, their respiration rate probably goes down, and that doesn't mean that all the organisms in the pond are sleeping at night. But I mean, I could just make it like, it's just, I don't like that one. More organisms are respiring at night than during the day. I mean, how can I go with C if I didn't go with B? Right? They are like the same answer in a certain way. Photosynthesis produces more oxygen than is consumed by respiration during the day. Well, if that's the case, then the amount of oxygen in the water should increase while photosynthesis is happening during the day. And then, of course, at night, when there is no photosynthesis, that oxygen will get consumed and it will decrease, which is the pattern that I see on this graph, meaning that D is the correct answer.
You got it.
Boom.
Hey, did you know that Teresa Barton wants to say a shout out to her AP Bio class?
I didn't. Would you like to tell us what that shout out is, young man?
Well, they are apparently the Honey Badgers. So nice. It says, "Go Team Honey Badger, you got this." So, considering what I know about Honey Badgers, I believe they probably do have it.
Yeah, I would think so. Absolutely. Over there at Pikeview High School, I mean, I would probably teach that class in protective gear, if that's the case. Bob, let's take a look at number 18, man.
Yeah, so this has actually come up a lot lately in terms of how you go from a chart to, some sort of phylogenetic tree. What you have to sort of approach this with is just looking at which of the species have the most similar patterns. So for me, this is more of a pattern recognition problem than anything else. So we've got 4 cladograms to choose from. The thing I immediately know is that Y doesn't have any similarity to anything. So I'm looking for a pattern that has Y out on its own somewhere. So that's going to get rid of B and D. Um, so that leaves me with A and C. And if I look at V and W, they're pretty close. They have one difference. And if I look at X and Y, or sorry, X and Z, they also have one difference.
Yep.
So I would expect them to be close together. And so that's what leads me to A, since C has them rearranged in a way that the table doesn't work. So
Yeah, I totally agree, right? It's just process of elimination way of thinking about this, right? Who have the most traits in common, and work our way through there. We can see that Y has none of the traits, so we would expect Y to be off on its own. And just looking at our patterns here, we see that V and W have more traits in common, and we see that X and Z have more traits in common. So I'm looking for a pattern on my tree where I see those grouped up. And once I find that, and helpfully it's the first answer, so I can just rule my way through it and move on.
Right.
Looking at number 19, we have a laboratory investigation, probably similar to what a lot of people out there have done. We put a solution of starch and glucose into the dialysis bag. And we see that we start with the starch and glucose in the bag, and the bag is in water. And then over time, the bag swells up. We find glucose in both the bag and in the water. And we only find the starch still in the bag. And of course, we find water probably in both places. The investigation is aimed at understanding how molecular size affects movement through a membrane. Which of the following best represents the amount of starch, water, and glucose in the dialysis bag over the course of this experiment? Well, the starch didn't move, right? It stayed in the bag, which means it remained constant. So I need to pick one where starch is constant. And so the starch is the little dash line. I need a little dash line to just be a straight line. So that instantly rules out C and D for me. I need A or I need B. So then if we're looking at what happens in each of these, we can see that in the bag swells over time. Well, why is that? Probably because water moved into the bag. So now I'm picking one where the water line is increasing over time. So that gives me A. I'm good.
Yeah, and even if you don't make that connection,
do the glucose.
It's the glucose.
Right. The glucose goes down over time because it moves out of the bag. You're still stuck with A. You cannot leave A.
Right.
Bob, you want to work us through this next set?
Yeah, sure. So this is a pretty famous classic example. Does it even say that? It may say that up there. But, I mean, this is a sympatric sort of speciation type of example where we've got hawthorns and apples. It's a long reading passage, and I'm not going to read it. But
so you could probably be like, if you've never seen something like this before, you'd be like, how am I supposed to read this if I only have a minute per question? And you do have 3 questions that you need to answer, but you probably do not need to spend a minute answering each of those questions. So you have time in the buffer in terms of the reading. So, hopefully you're able to read that. You can take a minute to read it, right? And then answer the 3 questions in the other 2 minutes or so on. Or maybe here's where you say to yourself, you know what? I know I used a rule of thumb as a minute per question, but that was to give me a little bit of extra time at the end, right? And so maybe you just spend a little bit more time so you can pick what you need in order to do it. There are other reading strategies you can use, but I feel like a lot of those are also, they're just going to eat into your time. You can definitely do them, and certainly, Bob and I are not experts in how your brain works. So if you have a method that works best for you for reading a question series like this, then you need to attack it from that perspective.
Right. So if you look at the first question, number 20, the divergence of the populations must have happened quickly because, well, some of these answer choices are just outlandish. So, gunpowder, I'm immediately just going to ignore that. Rail transport, I'm going to immediately ignore that. And then flies were imported. Well, no, the fruit was imported.
Right.
So the only answer that works is A.
Yeah, you can get rid of flies were imported by reading the first sentence of this reading, right? Native to North America. All right, so the flies weren't imported.
So, and then for 21, you're looking at some sort of zygotic mechanism here. So, which is the most important to prevent gene flow? And you have to go to the the third paragraph to find this and why it's temporal isolation, because it says that the apples set fruit earlier than the hawthorns do, that the hawthorns are later, and that's a difference in time. So you have to extract it from there. And then for the last question, you're looking at hybrids that can reproduce, but when they reproduce, they're the offspring of those hybrids are not very good, and their fertility is reduced, so it has to be C. So 2 zygotic questions, and then one, that first question should just be an easy one to get. So
Yeah, nice. Cool, man. Looking at 23, we've got 2 graphs of mouse color. And we seem to have like before, and then they give us some information about the release of the mice, right? And we see that before the release, or right after they released them, most of the mice were brown. Yeah, higher frequency of brown mice. But then 20 years later, it seems like that shifted. We now have more gray mice than brown mice or black mice. They give us information about what they did and how they measured this and so on. And then it says to us, which of the following best explains the change in frequency distribution of the fur color phenotypes in the mouse population is shown in the figures above. So the allele for gray fur color is unstable. I don't really like that term when describing alleles. It's not a term I've ever used. The stability of an allele. I mean, I can kind of see where you might get that in your brain. But, that's okay, kind of. But then if you look at the second part, right? Most of the alleles mutated to become alleles for black fur. That is not how this dance works, right? That's just not how biology functions. So it's kind of a nonsense answer. The field was composed primarily of light-colored soil and little vegetation. Well, if that's the case, then the gray mice would have protection from predators. We would expect that they would survive and reproduce, pass on their gray color alleles. We would expect over time we would see more gray mice. That makes sense to me. That seems like it makes sense from an evolutionary perspective. It's the logic there seems solid. Looking at the other ones, sexual selection lead to increased mating frequency of black and brown versus gray and brown. I don't have any information about the inheritance modes of these patterns. I don't have any information about sexual selection here. I just don't like it. And then the gray mice were hardest to capture, and so were underrepresented in the 20-year sample. Well, there are more of them than any other color. So even if they were underrepresented, that just means that there's even more gray mice, which means that they're at an even greater advantage. So that does not, the logic of the quantitative thinking there doesn't really seem to work out. So I think we're good.
Hey, Miss O'Reilly from Mamaroneck High School.
My little neck of the woods, Mamaroneck High School, a little upstate from where I am right now.
Oh, yeah?
Yeah.
That's in New York?
It's in. I'm assuming it's the Mamaroneck in New York. How many Mamaronecks are there?
Is that Long Island?
It is not. It is slightly upstate.
Oh, okay.
Slightly upstate. I apologize, Mamaroneck, because you probably don't consider yourself to be upstate. But you've got a Long Island boy here, so just remember, like, literally everything is upstate from me right now.
All right, for 24, we've got some amphibians. They lack genetic diversity. They're separated from each other by wide areas of dry land, so they must be in some sort of ponds or something. So which of the following human actions is most likely to improve the long-term survival of the amphibians? So we're looking at something that's going to help them. Cloning the largest individual to counteract the effects of aggressive predation. There's no guarantee that cloning the largest individual is going to have any effect on predation. So I'm going to get rid of that one.
Sure.
Reducing population size to decrease competition for limited resources.
I think we're working in exactly the opposite way we want to be working.
Right. So that doesn't make sense.
Not a well regarded conservation habit is to actively reduce the populations of genetically endangered species. Just not a way that usually we go about the process.
Yep, and then making a dam to control flooding. I think you'd want more water, not less. So that leaves D, where you're building ponds to promote interbreeding between the population. So you're increasing gene flow there. So I would say that that's correct.
Yeah. I would also point out with the cloning, it's talking about a lacking of genetic diversity being the problem. And so if you clone everybody, they're all going to be genetically identical, which doesn't seem to solve your genetic diversity problem. But I totally agree. Absolutely. It's got to be D. Cool, man. 25, a new mutation arose in one copy of gene X in a somatic cell, which resulted in the formation of a tumor. Which of the following pieces of evidence best describes how the new mutation directly caused the tumor? So what do we know about tumors? We know that tumors come about as a result of uncontrolled cell division. We know that cell division is largely controlled by basically two major classes of genes. You've got your tumor suppressors, and you've got your oncogenes. And so as a result, let's see what we got going on here. Protein X normally stimulates cell division, and the mutation creates an overactive version of Protein X. So now the thing is not normally stimulating cell division, it's crazily stimulating cell division. That seems like it might lead to cancer. You know, it wouldn't be the only thing that had to happen, but it would definitely set us on the road to a tumor, certainly. Protein X normally activates a growth hormone receptor, and the mutation decreases the stability of Protein X. Well, growth hormones, right, are the things that cause cells to divide. And so if we decrease the stability of the protein receptor, we're going to decrease the reception of that message. That would lead to less division. Protein X normally prevents passage through the cell cycle, and the mutation creates an overactive version of Protein X. Those cells would just never divide again then, right? They'd just be stuck there forever because that's what an overactive version of that protein would do. Protein X normally regulates gene expression, and the mutation created an underreactive version. It's the same issue again, right? If it's underreactive, it's not going to drive cell division here. So as a result, choice A is the only one that logically makes sense. You don't even need to know that choice A is basically the definition of an oncogene in order to figure this out. You can just puzzle your way through it just by thinking about the effects of these mutations on the overall likelihood of the cells to divide.
Yep. Just some logical thinking goes a long way on these tests.
Big time.
For 26, this is another kind of classic example. It's been used a number of times. So we've got our cystic fibrosis model here. Without getting into too much depth, we've got a mutation, and the mucus layer that normally presents on the surface becomes dehydrated and thick. So the question is, an answer to which of the following questions would provide the most information about the association between the CFTR mutation and the viscous mucus. These kinds of questions are becoming more and more common, where you're picking, which of the following claims, which of the following ideas are the best. And my students really struggle with these because they're not.
I think everybody does.
Yeah. So. And a lot of times the choices are fairly similar, so it's a little bit more difficult. So is the mucus also secreted from cells through the proteins? Well, that's not really satisfying what we want to know. We're looking for the association between the mutation and the viscous mucus. So plus the mucus isn't being secreted through those proteins. Most likely we're seeing chlorine ions being secreted. So that one doesn't work. How does the disrupted chloride movement affect the movement of sodium ions in water? Well, that might work because we're looking at viscous mucus related to the mutation. So I'm going to say that's a possibility. How does the mutation alter the structure of the proteins? Well, the protein structure is not what's sort of on trial here. It's the relationship between the mutation and the mucus. The structure of the protein, that's a tough one. So I'm not going to rule that one out yet. And then what's the change in the nucleotide sequence? Well, that has nothing to do with
unrelated.
We're being asked. So we're really down to B and C. And to me, between those two, we're looking at the association between the mutation and the mucus, the viscous mucus. What's causing that is the transport problem. So I would go with B on that one.
Yeah, man, absolutely. But these are tricky, right? These are the ones that I think most of our students definitely struggle with. Yeah.
Yeah.
Hey, Bob. Folks out there are probably wondering how we're doing this, and you are calling into me through Google Hangouts, right?
Yep.
And then, of course, I'm streaming from my desktop onto YouTube. Both of these products are, of course, Google properties, right, Bob? And as you may know, Google is, of course, seated in Mountain View, California. Ms. Danielle Schroeder writes to us from Mountain View High School, I'm assuming in Mountain View, California, home of the Mountain Lion Gladiators, and would like to say that Mr. Fluffy and I believe the force is strong with each and every one of you, by which I assume she means her students. Go black and silver. So I've either just really ticked off a different Mountain View somewhere, or I've repped the local major business concern as strongly as I possibly could.
And we do know that since they are black and silver, that they are on the dark side of the force. So
cold, man. Cold.
They've got that going for them.
27 through 31 are another one of these series. So what do we got here? We've got an experiment from the 70s investigating gene expression. We have a lot of information about it up front. Probably going to be a good idea to read this information. But we take rabbit hemoglobin mRNA, we put it into eggs. The eggs grow up into tadpoles. They cut open the tadpoles. They dissect them. They look at the different tissues, and they look at the amounts of frog tubulin, and then rabbit beta hemoglobin and rabbit alpha hemoglobin, which of course was a product that was made as a result of the injection of the rabbit hemoglobin mRNA. So looking at these questions here, the observation that the rabbit mRNA was successfully translated into frog tissues supports which of the following conclusions? Frog cells are able to replace their own hemoglobin with rabbit hemoglobin? Well, not necessarily, right? They're just making the protein. I don't think they're necessarily replacing their own, right? Undeveloped frog eggs can be induced to form genetically identical copies of a rabbit. I see nothing about that here in the question. Rabbit hemoglobin can induce an immune response in frogs. I see nothing about that in the question. Rabbits and frogs share a common genetic code for expressing heritable information. If that was the case, then the gene from the rabbit, when given to the frog, would be no problem for the frog's ribosomes. They would be able to translate that protein without a problem, and we would expect to see the rabbit protein in the frog tissue, which is exactly what we see. So choice D is the correct answer for 27. Looking at 28, the electrophoresis result best supports which of the following conclusions? Cell specialization during development results in some cells losing the ability to synthesize proteins. That's probably a true statement. I don't see anything about that here, right? Everything looks the same based on what we're looking at here. So I'm not going to conclude that based on these results. Cells from different tissues share a common ability to use genetic material from a foreign source to produce protein. If that's true, then the injection of rabbit hemoglobin mRNA into the eggs is going to lead to the expression of those genes in all of the tissue. And that is what we see. That seems like the correct answer. Let's look at the other ones. In comparison with other cells, nerve cells have a superior ability to produce cytoskeletal proteins. They tell us in the paragraph that tubulin is a part of the cytoskeleton. But we see the same amount of tubulin in both types of tissue. So that doesn't seem to work for us. And muscle cells produce more beta hemoglobin than do cells from the other tissues in a tadpole. No, it looks like they're making the same amount of rabbit beta hemoglobin. I don't know how much native hemoglobin or frog hemoglobin they're making because I don't have that information. So B has to be the correct answer here. 29, which of the following is the best justification for why the rabbit hemoglobin proteins were found throughout the tadpole? Rabbit mRNA is composed of nucleotides that are more stable than those in frog mRNA. I don't have any information about frog mRNA stability. Cannot conclude what you do not have information about. Rabbit hemoglobin is synthesized more efficiently than frog hemoglobin. I don't have any information about frog hemoglobin production, so I cannot conclude that one either. After differentiation, the rabbit hemoglobin proteins move through the circulatory system of the tadpole to every cell. That's just not how things would work here, right? The proteins would be made in the cells, right? The mRNA would be present in all of the cells that come from that original egg cell. And so it would just hang around, and every cell that was produced from that progenitor egg cell would just translate the rabbit hemoglobin. So that doesn't work either. The mRNA is injected in the newly fertilized frog eggs. It's distributed in the cytoplasm of every daughter cell during cell division. I just explained how that would happen, right? But if we inject it in there, and then that one egg cell gives rise to every cell in the organism, every cell in the organism is going to have some amount of this rabbit hemoglobin mRNA. So D is the correct answer for number 29. Looking at number 30, which of the following conclusions is most consistent with the results of the experiment? Rabbit mRNA is composed of nucleotides that are absent from frog mRNA. Show me the data. Where is the data? I do not see the data, so I will not be concluding that as my answer. The larger volume of blood circulates through a rabbit than a frog. Probably true, right? Seems like it's true. I have no information about that here. It's not related to the experiment. I am not going to choose it, right? The subunits of hemoglobin differ in size, shape, or charge. Well, if this is the case, then when we put them on the gel, they should separate out into different sized bands, right? We should get bands in different locations on the gel. And if we look at the data of the gel, we see a band for beta hemoglobin and a different band for alpha hemoglobin, which is exactly what we would expect if C was true, which is why it's the correct answer. Synthesis of beta hemoglobin occurs at a faster rate in muscle cells than in other body cells. No data to conclude that, right? So here's an example where 3 of these answers, maybe they're true, maybe they're not. B is probably true. I'm pretty comfortable concluding that most rabbits have more volume of blood than frogs do. At the same time, I have no data about that here. And I'm sure I could pick a really big frog and a really tiny rabbit in order to make a counterexample. So it's not something that you can pick. Looking at 31, given that equal amounts of the different mRNAs were injected into fertilized frog eggs, which of the following conclusions is most consistent with the electrophoresis results? So looking at this, we see on the gel that we have a bigger band of beta hemoglobin than of alpha hemoglobin. But we injected the same amount of those hemoglobin mRNAs into the original cell. But yet these cells are preferentially making more beta hemoglobin than alpha hemoglobin. So why is that? Beta hemoglobin is translated more efficiently than is alpha hemoglobin. Doesn't seem terrible to me. I'm gonna leave that one up there. Alpha hemoglobin is present only in cells where beta hemoglobin is absent. I don't think that is really abides by the data. I'm getting both hemoglobins in both cell types. Alpha hemoglobin mRNA is more stable than beta hemoglobin mRNA. If that was true, I would expect to see more alpha hemoglobin than beta hemoglobin. I do not see that here. Tubulin inhibits translation of hemoglobin. If that were true, then anywhere tubulin was being made, no hemoglobin would be being made. So I have eliminated B, C, and D. The only one that makes any sense is A, and that's the correct answer.
Very good.
I just smoked that series, Bob. Right?
Yeah.
I've now got all this extra time that I can use for other things as I go through my exam here on my first time taking an AP exam.
You know, with our last 2 names, it's kind of a shame to have Josh Farr say his kids are far out AP Bio. You know, we really can't use our names to do that sort of thing. So I'm a little jealous.
Yeah, it's nice, man. And double R's on the Fars, that's great.
So he's at Cienega High School?
It's either Cienega or Cienega. I'm going to go with Cienega.
Okay.
Why not?
And they apparently rock the flaviviruses. So they have that going for them.
Yeah, I mean, has they been sick?
I don't know.
God, I hope not.
I don't know. We'll leave it at that.
Bob, take us through number 32, man.
All right, so 32 is kind of irritating.
Oh, I remember 32.
And so you've got a phylogenetic tree that is going to have a lot of different sources of information binded, rolled into one to make this tree. And so we've got all these organisms, you've got the tree, and then the question is, which of the following states is most consistent with the tree shown? So with the tree shown. But then you have all these things that aren't given to you on the tree.
Right.
So birds and turtles evolved their own means of gas exchange independently of the other vertebrates. So the thing you have to read into here, if that's true, then birds and turtles should be next to each other, perhaps.
Right.
Birds and turtles are spread pretty far away from each other, so that's not going to be my choice. Mammals are most closely related to birds because they share a direct common ancestor. So all you have to do is look and see if mammals and birds have a common ancestor directly. And they should be on the same clade if that's true, like the
same group.
Right next to each other.
Yep. Yeah, right next to each other.
Monophyletic group, and that's not true. The common ancestor of reptiles, birds, and mammals produce amniotic eggs. So this is the thing. So they all are on the same clade. that's not given to you as information. So you have to sort of intuitively know, okay, that's a homology that they share. And that, I think that irritates some kids because it's not given. So I think, that's going to be the right answer. They are all in the same grouping branch, the way the tree's made. So that does make sense. The last one, crocodiles, direct descendants of ray fin fishes, doesn't bear out. So they're on totally two different parts of the tree.
Yeah.
So C is going to be the correct answer, but I'm not going to say it's a bad question, but
Nope.
It is a challenging question.
It is a challenging question, and you can get your way to C if you know enough about phylogenetic trees to eliminate A, B, and D. For the reasons that Bob just explained, right? If birds and turtles evolve their own means of gas exchange independently of everybody else, they should be next to each other. They're not. So it's a wrong answer, right? If mammals were most closely related to birds because they shared a direct common ancestry, they'd be right next to each other. They're not. So it cannot be a correct answer. And if crocodiles were the direct descendants of ray-finned fishes, they would be right next to each other. And they are not. So as a result, right, just by tree knowledge of how trees are built, you can be left with C. Does it help you if you know that the common ancestor of reptiles, birds, and mammals produced amniotic eggs? Yeah. I mean, it's super helpful at that point because you have that piece of knowledge in your brain. But different teachers are going to have taught that or not taught that. And I, to be perfectly honest, haven't required my students to know that for probably half a dozen years now. So, it's a tough question for that reason.
Plus, the tree is kind of awkward looking, too.
Yep.
So. Anyway,
Nice.
Whatever.
Let's take a look at number 33. A student in a biology class crossed a male Drosophila melanogaster having a gray body and long wings with a female having a black body and apterous wings. Following distribution of traits is observed in the offspring. So we've got two traits here in the cross, right? Okay. So let's see if we can figure out this. Which of the following is supported by the data? Gray body and long wings are dominant. If that's the case, everybody should have gray body and long wings. Right? No, maybe not. Right? We could probably figure out some Mendelian ratios and see what that might look like if we had like heterozygotes. But then I think we'd expect 50-50 or 25-25, 25-25 in this tree. And I don't see numbers that are anywhere near that. The alleles for gray body and long wings are recessive. Gray body and long wings, it seems like those show up an awful lot in our offspring here. Maybe, I don't know, let's think about this. I could probably work out the cross there too. What would be helpful for me is if I remembered some of the kinds of ratios that I would expect to see if these things were inherited independently from each other, right? I would maybe, if it was two heterozygotes being crossed with each other, and it's not here, right? But I would expect to see a 9-3 to 3-1 ratio if they were inherited independently from each other. If I had a heterozygote and a homozygous recessive, right, I would expect to see a 25-25, 25-25 combination here. I don't see that either. Really, none of the classic Mendelian ratios are represented here. So as a result, that clues me in that something else is going on here. It's not just a classic Mendelian situation. And so as a result of that, we got some sort of linkage here. Yeah. So the genes for the two traits are located on two different chromosomes. Well, that's not linkage. So C is no good. So I'm really left with D. They're located close together on the same chromosome. And the reason we're getting those small numbers of gray body apterous wings or black body long wings is because of crossing over occurring between the two gene loci. Right? Those are recombinant classes, and we get small numbers of recombinant classes whenever we do linkage crosses. So just by a little knowledge of the ratios, I can eliminate the idea that it's just a classic Mendelian situation.
Good job.
Boom.
Boom. Hey, Jessica Davis, or we know her as Mrs. Davis. Her AP Awesome crew is in Austin. I'm assuming that's in Texas. At Bowie High School, which even gives me more evidence that it's in Texas.
It'd be really weird if it wasn't.
You all make me proud every day. I believe in you. So does the pink fairy armadillo.
That is the cherry on the Texas sundae right there.
They have the pink fairy armadillo.
Nice.
All right, so 34 should be a fairly quick one. We've got if you know the reflex arc, you've got a stimulus and a response, And just even by following the pathway, I think you can figure this out. The question is saying the arrows show direction of impulse transmission along the labeled axons. If axon 2 is damaged, another AP College Board, let's break it.
Yep.
If the axon 2 is damaged before the pinprick, which of the following is most likely? So if it was damaged before the pinprick, the person will not feel the pinprick. You're going to feel the pinprick. The person can no longer feel pain. I don't think that's going to be true. The person's finger will not withdraw reflexively. Well, if you look at where 2 is, that's going to the response, which is going to be the reflex. So that's where it's going to be broken. So you wouldn't have the reflex of withdrawal. And then the person cannot transmit nerve impulses to the brain. Well, the brain's not really, well, it is involved secondarily, but not with the reflex arc. This is happening before the brain figures it out. So I would say that C is probably the right answer, and move on.
Yep. Looking at 35, if chemical signals in the cytoplasm control the progression of a cell to the M phase of the cell cycle, then fusion of a cell in G1 with a cell in early M phase would most likely result in what? So we're taking these two cells, smushing them together, right? And so we have chemical signals present in that M phase cell that are now present in our smushed together fused cell. Replication of the chromosomes only in the G1 cell? Why? The chemical signals would probably trigger no replication, right? Replication happens in S phase of interphase. So that doesn't make a lot of sense to me. Exiting of both cells from the cell cycle and into the G0 phase, but those chemical signals that are controlling progression of the cell in the M phase, right, are now in both of those cells. So why would we go into G0? That doesn't make a lot of sense to me. Condensation of chromatin in preparation of nuclear division in both cells, that seems like a fundamental early stage mitosis thing that happens. And so if we now have those chemical signals present saying, hey, let's go into early stage mitosis, right, then that's what's going to happen here. So C is definitely our correct answer. Transfer of organelles from the G1 cell to the cell in the M phase. They're both the same cell now, right? They're smooshed. So as a result, you're not transferring things from one cell to another. You smooshed them together and made some sort of hybrid Frankencell. So that's what's going on. Choice C is the correct answer.
That's right. For 36, I'm going to do this in 10 seconds.
Boom.
Okay?
I'm going to time you. Ready?
Which of the following models depicts a non-specific response? There are no B cells, T cells, or in the non-specific response, so it's got to be B, macrophage.
Whoa! Whoa! Talent.
Take a lot of time.
Talent from Mr. Kuhn. Right there. Let's take a look at number 37. In the Arctic Ocean, we've got a lot of information about phytoplankton. In years where there's more open water, there's more zooplankton and fish than in years with less open water where there's more ice coverage. Based on the graph above, the difference is most likely because of what? Right? Number of months with open water goes up. The annual primary productivity goes up, which means the ecosystem is more productive, which means that it should be able to support more zooplankton and fish. So when there's less open water, light is blocked from the zooplankton, so they cannot... No, zooplankton don't care about light. Zooplankton are animal-like in their feeding patterns. They just want to eat something. That thing cares about the light, right? That's our phytoplankton. When there's more open water, the temperature is warmer, so the zooplankton and fish populations increase in size. I don't know. I don't know that warmer water makes these things increase in size. I have no information about temperature, so no, I don't like that at all. The ice blocks the light, so in years with more ice coverage, there's less photosynthesis than in phytoplankton. Well, that would seem to decrease productivity, and so as a result, that would then lead to fewer zooplankton, which would then lead to fewer fish as a result. The ice increases the light available for photosynthesis. That's exactly backwards, right? Then we'd expect more zooplankton and fish when there was more months with ice or fewer months with open water, right? Further to the left of our little graph here. So, in fact, choice, what did we say? C is the correct answer.
Good. Miss Hunter, or Mrs. Hunter's AP Bio class, Rocky Point High School.
Rocky Point High School? Right down the road, babe. Right down the road. Rocky Point, Long Island. I would say that Rocky Point, Long Island, arguably further downstate than where I am right now.
Well, did you know she has a lot of fun puns here? What's stomata? A lot of fun things have transpired this year. And you've shown to have great potential. Good luck.
Nice job. Nice job.
All right, 38, we've got a eukaryotic gene expression problem here. We've got everything linked up here for the transcriptional start of gene X. When the expression of gene X is activated in liver cells, previous studies have shown that gene X is never expressed in nerve cells. Based on the diagram, which of the following most likely contributes to specific expression pattern of gene X? So, A, expression of gene X produces large amounts of tRNA, but undetectable amounts of mRNA. We have no information based on the model that could be true. So we're going to get rid of that one. The general transcription factors inhibit the activation of gene X in liver cells by blocking the activator from binding to RNA polymerase II. So the transcription factors inhibiting the activation of gene X in liver cells by blocking the activator. So we'll keep that one on the table for now. The activator is a sequence of specific DNA binding protein that is present in some tissues, but not in other tissues. That makes sense because the STEM said that we've got expression in liver cells, but not in nerve cells. So I want to keep that one. And then D, the enhancer is a unique DNA sequence that's added to the nuclear DNA in some cells during the process of mitotic cell division, but not other cells. It doesn't make any sense.
That's a Nobel Prize winning discovery right there. I've never heard of that.
Right. So we're down to B and C. I think C is probably what I would choose as the correct answer. David, do you have any reason you would automatically get rid of B?
Well, C makes very clear sense, right? And it's a good, clear explanation as to why it might be expressed in one cell but not in another, particularly based on the image that we're given. If we look at B, the general transcription factors, right, which we see here, inhibit the activation of gene X in liver cells by blocking the activator from binding to RNA polymerase II. It looks like those general transcription factors, right, need to bind. They're the things that need to bind to RNA polymerase II in the picture to lead to expression of gene X. So the description that they're giving us here of them inhibiting the activation of gene X is backwards based on the image that we see here.
Right. So you have to go to the model and take a look, and you see that that's not what they show. So,
Bob, I'm going to go ahead here and do numbers 39 and 40.
Whoa.
I know. I'm going to mix it up. So looking at 39, the diagram above illustrates feedback control is exerted by the hormone thyroxine. Following surgical removal of the thyroid gland, the level of TSH in the blood will increase. Which of the following best explains this increase? So we've removed our thyroid gland, so we now have less thyroxine as a result. We have less thyroxine, which means the inhibition of our anterior pituitary by thyroxine goes away, right? Which means that our anterior pituitary is now going to increase the amount of thyroid stimulating hormone that it is making. Also, the inhibition on the hypothalamus goes away, so we'll get more TRH, which leads to more TSH as well. Bob, you may not see what I'm doing here, but I've actually just X'd out some things and drawn some arrows on my little diagram so that I can figure out what's going on. Residual blood thyroxine from prior to the thyroid gland removal will bind to cells in the anterior pituitary, signaling more TSH secretion. No, because that is not what thyroxine does in the anterior pituitary. It inhibits it, and besides, the gland is going away. There's not going to be too much residual stuff in the blood. Thyroxine will remain bound to thyroxine receptors on various body cells, and these cells will secrete additional hormones that stimulate the anterior pituitary to secrete TSH. No, we can get our answer much more clearly just by looking at the feedback loop that I've just put my X's and my arrows in. Thyroxine was stored in the anterior pituitary prior to what now? Thyroxine was stored in the anterior pituitary. That's not a thing that happens in the diagram, right? The thyroxine's made by the thyroid gland. So that's just wrong based on the picture. A decrease in thyroxine levels means a loss of inhibition to the hypothalamus and anterior pituitary. Those are those X's I just drew where we had the inhibition and leading to increased TSH secretion due to the arrows that I drew. So I think D is the correct answer for that one.
Very good.
Number 40, we have some information about tasters and non-tasters. The populations are in Hardy-Weinberg equilibrium. The allele for non-tasters is recessive. In which population is the frequency of the recessive allele the highest? Man, I know a lot about Hardy-Weinberg. I might get out my equations and start doing it if I didn't stop and think for a moment. I've got non-tasters. I know that they're Q squared in the equations, right? Because they're the recessive condition. So the population where the number of individuals who are Q squared are the greatest frequency of the total number of individuals in the population, that's going to be my winner here. I could get my calculator and calculate it out if I wanted to, or I could just estimate. 32 out of 142 is approximately 25%, right? 4,328 of 12,563 is approximately a third, or 33%. 500 out of 715 is 66% approximately. 2596 out of 14085. I don't really even know what it is, but I know that it's nowhere near 66%. C is the correct answer. That kind of speedy mathematical thinking is brought to you by Mr. Camden Hanslick Burton over at Summit Sierra High School, who would like us to know that his AP biology class is an incredibly dedicated group of Spartans who've worked tirelessly during school and after school at our weekly AP office hours to learn science and prepare for the exam. Especially to the 3 students who wanted to improve their scores from last year and came back for year 2, Bob. Year 2. I mean, that's an applause for those guys right there. But I'm sure at those office hours that Mr. Camden Hanslick Burton taught them that kind of mathematical reasoning that allows us to conclude C and move on without wasting any more time on that question.
No, because you needed that time because now you have a reading question you got to do.
Correct.
41 to 45. So this is based on an old AP lab that you do. Here we have a electron acceptor that's very similar to other electron acceptors like NADP+. And so this DCPIP is a dye that can accept electrons from the ETC. And the transfer of electrons to this molecule decreases the relative absorbance of specific wavelengths of light, 605 nanometers by a solution that contains the dye. So we're going to be looking at the dye color changing. And what they've done is they've taken this reaction with a buffered solution with chloroplast. They've made a bunch of samples, they put them at different distances from a lamp, and then they're going to measure the absorption, I'm assuming in some sort of spectrophotometer, and they're getting some data from that. So they have sample 3 is totally covered and wrapped in foil. So we have sort of a dark situation there. So 41 says, which of the following provides the best indication that light is required for the activation of electron transfer reactions in chloroplasts? Okay, so the thing here is that light is required. So if you need to know if light is required, you need to be able to compare it to something. And the thing you need to compare it to is when light isn't required, which is in the dark. So the only answer choice there that compares dark and light would be B, comparing 2 and 3. Okay, that's what I would choose for that one. Which of the following can reasonably be concluded by the experimental result? So reasonably concluded. So we've got A, chloroplast must be suspended in a buffer solution to function properly. We know that there was a buffer solution, but we don't know that they have to be. So I'm going to get rid of that one. The optimal temperature for the activation is 25 degrees. We have no information on temperature. We assume it's controlled, but who cares because it's not a factor here. The DCPIP inhibits biochemical reactions in suspended chloroplasts. Well, no, there's a reaction that's actually taking place. So we know that that's probably not it. And then light from the lamp can substitute for sunlight in simulating chloroplast processes. It's a goofy answer, but it's the right answer, I think, here. They are using a lamp, and it is part of the experiment. It's a very non-technical sort of answer choice, but it is the answer choice for that one. If we take an additional sample containing the chloroplast DCIP solution, and we're going to place it further away, so we're choosing a prediction. of what the result would be. So the concentration of the DCP-IP will increase. It should decrease because of the increased time it's going to take to react. B, the absorption at 60 minutes will be roughly equal to 1.4.
1.4.
How do I know that? So I don't know where they came up with that. So I'm going to say when they commit to a value like that, I'm going to say no.
Could we ever get an absorption higher than 1? I don't think we could.
Not unless your machine.
I certainly don't see it in the data.
The change in absorption or absorbance over time in the solution will be less than that of the other samples. Makes sense if you have a progression. I would suspect that that's the right answer. And then the D is just a red herring. There's no reason to believe that temperature would increase. So we would go with C. Which of the following descriptions of photosynthesis best explains the results? Availability of electrons for transfer to DCP-IP depends on light energy. That's probably what I'm going to go with, but let's look. Movement of this DCP-IP across chloroplast membranes occurred in less than 60 minutes. I don't know that it has to be less than 60 minutes, and so I don't think that's going to be a good choice. C, chlorophyll molecules degrade rapidly in the presence of DCP-IP. The chlorophyll molecules aren't degrading, it's the DCP-IP that is changing. And then lastly, DCP-IP can only be used to measure photosynthetic activity at low light levels. Well, you know, I don't think that that's true. We have a wavelength of light of 605 nanometers. That seems to be doing the trick, and plus I don't know that that's true, so I'm going to say that A is the right one for that one. My thought process, you may have a different one, but and then lastly, which of the following scientific questions could be investigated using a similar experimental setup? A, carbon dioxide has nothing to do with this because these are the light-dependent reactions. I'm going to just move on by that one. B, what wavelength of light best activates the electron transfer reactions in chloroplasts? I think that's pretty reasonable, but I'm going to look at the other two. Which molecules in chloroplasts accepts activated electrons from DCP-IP during photosynthesis? I don't know that DCP-IP is going to give up. Which molecule in chloroplasts accepts activated electrons? I don't even know if I know what that means. So I'm going to skip that one. And then D are the same genes that are expressed in chloroplast. That's a red herring. We're not comparing chloroplasts to mitochondria. So I guess I would be down to B and C, and B seems like the most logical.
Yeah, I mean, I think we could look at this experiment and see how we could do something very, very similar, but instead of varying the distance from the lamp, right, we could probably just vary the colors of the light that we used. Right. And so it would still work very much in the same way. The only thing we would be changing is our independent variable. Nice. Good series, man.
Yeah, kind of exhausting, but yes.
Let's go on. Let's take a look at number 46. And let's do that for Dr. Close over at Hickman High, who would like everybody in her AP Biology class to enjoy prom, Cupies, and remember epigenetics. So, 2 small requests from Dr. Close over there at Hickman High. The figure above, this is number 46, shows a model of a ligand precursor being cleaved to produce an active ligand that binds to a specific receptor. Which of the following is most likely to reduce the binding of the active ligand to its receptor? Change in the cytoskeletal attachment of transmembrane proteins, it has nothing to do with the binding of the ligand to the receptor. The presence of a large amount of the precursor form of the ligand has nothing to do with the binding of the active molecule to the receptor. An increase in the ratio of the number of unsaturated, the number of saturated fatty acid tails in the membrane lipids, nothing to do with the binding of that molecule to the receptor. Right? These are all things that don't have to do with the mechanism that's at heart of this question, which is the binding of that ligand to its receptor. That's a protein shape thing. None of these answers are affecting protein shape. However, D, a mutation in the receptor gene that causes the substitution of a charged amino acid for a nonpolar amino acid in the ligand binding site of the receptor. Well, if we change amino acid, particularly with a big chemical change, that's going to change the overall shape of the active. It's not even an active site, really, but it's kind of the same thing, right? It's going to change the shape of that receptor binding site on that receptor protein. And so as a result, that's going to maybe affect the ability of that ligand to bind to the receptor.
Hey, I was just thinking about Dr. Close in our last shout out. Enjoy prom cupies and remember epigenetics. Is it and remember epigenetics, or is Dr. Close implying that prom will have some sort of epigenetic effect on the kids, so they need to remember, don't do something because of epigenetics?
Maybe.
It just got me thinking.
Yeah, man.
Look, Dr. Milks, South's AP Bioheads. We know her. She just had her birthday recently, by the way, too. So DHS South, thank you for putting up with me. You guys are the greatest. No, you are the greatest, Kirsten. Let's go all the way in. So let's do this.
Very nice.
47. So this is kind of a sadistic experiment where we keep organisms in the dark and we don't feed them. Oh, wait, we do provide them nutrients over the 24-hour period. We do some mass measurements before and after. So which of the following is the best explanation for the pattern of change in mass of the organisms over time? So you've got 3 different organisms. You've got a plant, you've got a goldfish and a sea anemone, and all of them are aquatic. So evaporation's not going to be an issue. Conservation of matter, well, we've got mass changing, but that's not going to explain the change. And then growth and reproduction. If they're growing and reproducing when they're kept in the dark, we don't even know, are they by themselves? Who knows? So that's one I wouldn't go with. But any mass change that where they're able to respire, we should know that cellular respiration is going to cause a mass change. So that's going to be the right answer.
Nice. 48 to 51. So this is a complicated series of questions here. We're given a bunch of data. We're given a bunch of different types of data. We're given information about sickle cell, which of course I'm sure most of us out there know a lot about if you sat through an AP biology class. We're given some information about the inheritance of sickle cell. We're given a pedigree of a family with affected individuals. We're given a portion of the data from the wild type allele that codes for normal hemoglobin. We're given the codon chart. We are given the different fragments from different individuals on that pedigree. And now we're asked a bunch of questions about it. This is going to take some time. We're going to need to work our way through this. So let's take a look. Based on the data shown in Figure 1, which of the following best describes the genotypes of individual family members in the pedigree? All affected individuals possess at least one dominant allele of the hemoglobin beta gene. No, if you're affected for sickle cell, and it tells us this, right, you need to have 2 copies of the recessive allele. So that's not good. Healthy individuals may possess one mutant allele of the hemoglobin beta gene. That is certainly the case, right? That can absolutely happen. You, in fact, see it happening in individuals 7 and 6 with the child that they have. They're having an affected child, but they're both not affected, which means that they are carriers for it. That is the correct answer. Individuals 4 and 5 must be heterozygous. Individuals 4 and 5 are homozygous recessive. They both have sickle cell. Individuals 2 and 6 possess 2 copies of the wild type allele. Individual 2 does not possess 2 copies of the wild type allele because he is producing affected children, which means that he has to be transmitting his recessive sickle allele. So that's not good either. The correct answer here is B. Looking at 49, the HBS allele, which causes sickle cell disease, results from a mutation in DNA sequence shown in Figure 2 that produces a valine in the place of glutamic acid residue in the hemoglobin protein. Which of the following mRNA sequences is derived from the HBS allele? So in order to do this, I think the only way I can really do it here is to figure out, look at my template strand that's given to me in Figure 2. And so it's 3' GAC, which means that I'm going to need a 5' CAG. No, I'm sorry. Pardon me. GAC, I'm going to need a 5' CUG. Is that not correct, Bob?
You're right.
And if that's the case, I believe that D is the correct answer. I do not think I need to do anything else.
And I think by including the amino acid chart, that's such a distractor because you don't need it.
Right.
You know?
All I need to do is just make my transcript off of that template strand for one codon, Yeah, and check my letters like I did, and then you wind up with D.
Right.
Number 50, the restriction endonuclease MST2 recognizes the sequence, and they give us the sequence, right? 5' CCT, any nucleotide AG, and cuts DNA at that site, producing separate fragments. Which of the following best explains the banding patterns exhibited in Figure 4? So if we look at that wild type hemoglobin strand, do we have 5' CCT anything AG? We absolutely do. We have that site. It's the third and fourth codons on that site. On the non-template strand, we can see it pretty clearly. So it's going to cut it there. So if you're a wild type, right, if you have an HPA allele, that's going to get cut. And so you're going to have 2 fragments that are produced. Of course, if you're not wild type and you actually work out the codons that are produced here, you'll see that the substitution that they talk about in 49 is what causes that site to go away. That site is no longer going to exist. And so as a result, the enzyme will not cut, and you will only get 1 band, which you can also see because individual 1 only has 1 band, and we see that individual 1 has sickle cell. So which of the following best explains the banding patterns exhibited in Figure 4? The HBA DNA contains a restriction site for the MST2 restriction enzyme. That's absolutely true. We can conclude that based on what I just told you. You can also look at individual 3 and conclude that as well. The HBA-HBS DNA contains 3 recognition sites for the MST2 restriction endonuclease. It only has 1, right? It has the 1 site in that HBA alleles sequence. The HBS does not. So that is not correct. Individual 1 has only 1 copy of the hemoglobin gene. Nope, it's got 2 copies. They just both happen to be the HBS allele. So as a result, there's going to be no cuts to it. We're only going to get 1 band because they're both going to be the same size. HBS, HBA contains 3 different alleles for sickle cell disease. It's 2 different alleles, HBS and HBA. So no good, right? At this point, the AP exam is trying to wear you down because it's held you for 50 questions. And it's just trying to see if you can still hang with it. 51, possessing a single copy of the HBS allele has been shown to provide some resistance to infection by Plasmodium. Which of the following individuals represented in the pedigree would have the greatest selective advantage in an area where malaria is common? So we're looking for a carrier. Individual 1 has the disease. No good. Individual 2 is a carrier. That's the correct answer. Individual 3, we don't know if they're a carrier or not because they've only had 1 child who is phenotypically non-sickle cell. So based on the data we have, we can't think that they're a carrier at all. And then individual 5, right? Individual 5 has the disease as well. Can't go with 5 if you didn't go with 1. So the correct answer here is B. It's the only one that we know is a carrier for the trait. As a matter of fact, we know that individual 3 is not a carrier looking at the gel. Right? They only have 2 bands, which tells us that both of their alleles are the wild type allele. The set is over.
No.
It's not?
The set.
Yeah, the set is over.
Notice that there are 4 questions on page 33, but then 1 question on page 34. So mentally, they're giving you a little break of what to look at there. So we have a shout out to Miss Phillips AP Bio from Wheeler High School. And did I miss one?
That's all right. You can do Miss Phillips. We'll do the next one next.
Okay. But I think Miss Phillips gave us a lot of information there.
I think she just. Oh, I think she. Yeah, Miss Phillips told us why you guys are awesome, and it involved things like singing "All I Want for Christmas Is You," and "All I Want for Christmas Is a 5" to the tune of "All I Want for Christmas Is You." Miss Phillips, of course, walks around and gives you peppermints for your tests. You got each one to take home for the morning of the exam. So Miss Phillips sounds like a wonderful, nice lady.
I know.
Yeah, I want to be in her class.
I want peppermint.
I'm going to eat a peppermint on the morning of May 8th in solidarity with the students in Miss Phillips' class.
All right, so we're almost, actually, we're at the home stretch.
Yeah, man.
An art test, but
Boom.
There'll be more.
Yeah.
It was a real thing, so just hang in there. So we've got ADH, antidiuretic hormone. We've got a little information there about it, important for maintaining homeostasis in mammals. It's released by the hypothalamus in response to high tissue osmolarity. In response to ADH, the collecting duct and distal tubules in the kidney become more permeable to water, which increases water reabsorption into the capillaries. The amount of hormone released is controlled by a negative feedback loop. Based on the model presented, which of the following statements expresses the proper relationship between osmolarity, ADH release, and urine production? So the model is actually the paragraph. So that's what they're referring to. So what we have here are a number of answer choices that are just mixed up, and you have to figure out the right one. They're all the same kind of verbiage, but they're just rearranged. So A is tissue osmolarity rises, more ADH is released, causing less water to be excreted as urine. So that's going to be the correct answer. We don't want to excrete our water if we don't have to, and the antidiuretic hormone is going to prevent that from happening. So as osmolarity increases, less ADH is released. That doesn't make sense. As tissue osmolarity rises, more ADH is released. Okay, so far so good. But this causes more water to be excreted, and that's not what we want. The kidney is partially there to help us retain water when we need it, so it's an adaptation. D, as tissue osmolarity rises, less ADH is released, causing more water to be excreted, and that's not what happens. ADH prevents the water from being excreted, so A's got to be the right answer.
Yeah, man. Nice. 53 gives us a lot of information about Ellis von Creveld syndrome, and it tells us that in a reproductively isolated population, it's present in 1 out of every 500 live births, but in the general population, it's in 1 out of every 150,000 live births. It tells us to assume that both populations are in Hardy-Weinberg equilibrium, meaning that we can take those frequencies that they're giving us as gospel, right? And we can use that for the purpose of determining things. It says, which of the following best describes the difference between the frequency of the allele that causes the syndrome in the general population and the frequency of the allele in the isolated population? Well, if I know that in my general population, it's 1 out of every 150,000 live births have this trait, that means that the frequency of Q squared, right, is 0.0000667. That's what my calculator is giving me. The question is, the answers are all asking about the allele, so I need to take the square root of that. Right? So I just need to remind, and this is why they don't give you a scientific calculator on the exam, because you haven't looked at it in years, and you might forget how to take a square root. But I figured that out, I think. Nope, I got a syntax error. Fantastic. Doing it live.
It's 0.0025.
It's 0.0025 when I do it? Thanks. Yeah. Bob will not be present on your exam in order to help you do it. But so it's 0.0025. So if that's the case, it's either A or it's C, right? The bottom line here is that we have eliminated B and D as a result of doing that math.
Oh, wait, sorry. I didn't round. It's 0.0026. Sorry, David.
It's 0.0026. I don't really think it matters for the purpose of our... we did for the general population, right? So it's B or D. I take it all back. It's B or D. So let's look at the other things here. Showing that the rate of genetic mutation is highest among individuals in the isolated population. I don't know anything about mutation rates. Right? All I know is that this allele is higher at a higher incidence in the population. It's probably not the result of an increased mutation. It's probably the result of reproductive isolation. Reproductive isolation, right, is probably a good example of a phenomenon that we call genetic drift, which is what we see in choice D. So D is got the correct numbers, and it also makes the most sense, right, from a biological standpoint.
All right. Well,
that's it, man. We did part 1. No, because we have to do the grid-ins.
Wait, it's only been 2 hours.
It's an hour, and we've been on here for about an hour and 45 minutes. Let's talk about how we're going to do the rest of this. We're going to work through the grid-ins, and then we're going to lightly discuss part 2. But we're going to wrap pretty close to 9 o'clock. We'll talk about why we can do that with part 2 in a little bit after we work through the grid-ins. But without further ado, let's move on to the 5 grid-ins. Do you want to just work through each one?
Yeah.
All right, so why don't you do number 21? Just talk it out loud, and we'll work our way through it.
Okay, so this is a Hardy-Weinberg problem. So you're given the information that non-tasters of PTC are 45% of the population in North America. And so it says what percent of the North American population is homozygous dominant for the ability to taste PTC? And you've got to write your number between 0 and 1 to the nearest hundredth. So really, you got to realize first that we're talking about people. People are genotypes, so that's our 45% is going to be Q squared. If we work from there, we can take the square root and get Q. So Q squared is 0.45, Q would be 0.67. P is 1 minus Q, and that's going to give us about 0.33. And then if we square that, we're going to get P squared, which is what they're after, which is going to be 0.108.
And we need to give it as a number to between 0 and 1 to the nearest hundredth?
Hundredth, so 0.11.
Right.
And this is a range question, so if you had, I have the key here, it's also in your packet, but they would have accepted anywhere from, depending on rounding, 0.09 to 0.11, or 9 out of 100, or 11 out of 100.
So you could put it into fraction if you wanted. But
Right.
the best thing to do with a grid in is always to report your answer the way that the question is telling you to report your answer. Cool. That question was, of course, brought to us by Tyone over at Cruz with the AP Bio O'Gorman Knights, dedicated to excellence in attending the after-school lab sessions. Super important. Looking at number 122, based on the data shown, calculate the average rate of increase in oxygen consumption for animals acclimated to 5 degrees Celsius as the temperature increases from 10 degrees Celsius to 30 degrees Celsius. Give the answer in a big funky thing, but that big funky thing is actually just my y-axis divided by my x-axis, which is interesting because I have to do rate, which of course we know is change in y over change in x, and if we forget that, it is on our formula sheet. So we're going animals acclimated at 5 degrees Celsius. Those are the circles. We're going from 10 degrees up to 30 degrees. So I'm going up and figuring out what my y-axis is. I'm ending right about 190. I started at 80. So my change in y is 190 minus 80. It's 110. My change in x is 30 minus 10. It is 20. 110 divided by 20, I believe, is 5.5.
Wow.
I did that without a calculator. 5.5. So I'm going to take it to the nearest tenth. 5.5 is all I'm going to write down. Move on.
All right. 125 is really a graph reading.
Before we get to 123...
Oh, sorry, did I skip?
You, of course, wanted to tell us that 122 was brought to us by Allison Rice, Miss Rice's class at Magnolia High School, who wants her students, or wants us to know that her students happen to be the best Starbucks delivery service, as well as amazing students. She rarely has a morning without finding her favorite order on her desk. She would not have made it without them. That's awesome. I know.
Where does that happen?
And an entire nation of AP biology teachers start submitting their applications to Magnolia High School.
All right.
Mr. Kuhn, take us through number 123, please.
Yes. So this is a mark and recapture problem. Really, all you need to do is set up a proportion here. So it says the mark and recapture method assumes that the proportion of marked individuals in the recapture group is equal to the proportion of the marked individuals in the entire population. So knowing that, you can just set up your proportion for the part that's missing. So it says, using the results presented in the table below, estimate the total number of individuals in the population. Give your answer to the nearest whole number. So if you do this, you can take 14 over 180, and that's going to be equal to 88 over x. You do a little cross multiplying there. 14x is going to be equal to 15,840. X is going to be equal to 1131. You got it.
Nice. I actually did 180 over 14 is equal to x over 88. But you know what? I got the exact same answer because cross multiplying and dividing works the same both ways. Question 123 was brought to us by Cindy Carrick over at Hall High School. Hall High School AP Biology class, whom she loves with all her heart.
You got it.
Let's go through 124. A certain species of plant has 4 unlinked genetic loci, W, X, Y, and Z. Each genetic locus has 1 dominant allele and 1 recessive allele. For a plant with the genotype W, W, X, X, Y, Y, Z, Z, heterozygous at all 4 loci, what is the probability that the plant will produce a gamete with a haploid genotype of big W, little x, little y, little z? Give your answer as a fraction or as a value between 0 and 1 to 4 decimal places. So if I'm making a gamete and I can either go big W or little w, what are the odds that I would go big W? It's 1/2. And if I'm making a gamete and I can either go big X or little x, what are the odds that I would go little x? It's 1/2. And what about for the y? It's going to be 1/2. And what about for the z? It's going to be 1/2. Do I add all of those? No. Because they're independent events. And so the independent event law of probability of those all occurring simultaneously would be to multiply them by each other. 2 times 2 times 2 times 2 is 16. Put a 1 over it, it's 1/16, right? I would put my answer in as that fraction. I could also do 1 divided by 16, right? 1 divided by 16, and I'll get 0.0625, which is an answer between 0 and 1 to 4 decimal places.
Very good.
That question was brought to us by Karen Cunico over at Villa Walsh Academy. Her AP Biology 2017 Survivors. Animal crackers, $3 a bag. Yeast packets, $1 a package. A year of AP Biology together. Priceless.
Oh, that's interesting.
I like it.
Yeah. All right, so my favorite enzyme, you didn't know this, David, is phosphofructokinase.
Now I do.
Yeah, I know we all have our favorites, but that's mine. There's a lot given to you up there in terms of what we need to compare here, but in essence, it's going to be a graph reading problem. If I remember right, this had a pretty low mean because of something we'll talk about in just a second, but so we're looking at 0.5 millimoles. How many times more active is PFK in cells with 1 millimoles ATP plus 0.1 millimoles AMP than in cells with 5 millimoles ATP? So on your legend, it's kind of a obtuse kind of legend, but we can see our lines there. We got our x-axis. We're going to find 0.05 on our x-axis, and we're going to just basically draw a line upward. I just drew it through all the lines just to see where they intersected, but we're really going to be comparing the line that's dotted with the black dot and the line that is solid with the open circle. And here's the thing on this one. It comes out to be 0.5 and 5 or around that. A lot of kids are going to subtract that. Or they're going to... So basically, I think the mean was so low because kids weren't realizing that to go from 0.5 to 5 was a 10 times increase.
Yeah.
And I know when I saw this, when I talked to my students about it, they mentioned the fact that actually when I gave it as the practice exam, about half of them made that mistake and half of them got it correct. So just be careful if you're looking at your difference there.
That's definitely one thing that's going on here. I think the other thing that's going on here is that the legend is obtuse, right? It's very complex. The question that they're asking requires you to actually dial in and figure out what you're reading off of the graph. And you're at the end of the exam, which makes it really, really tough. And that's why John Gibney, over at TAISM, the American International School of Muscat, in his AP Biology class, would like you to know that there are 14 students in his class and he has 10 different passport home countries represented, all the way from Bahrain to South Africa.
Wow.
Mr. Gibney would like all of his students to know that it's been an honor learning with you this year. So that's the end of part 1. It took us about 2 hours to go through it, right? It's a little bit more than the time that you'll have to go through it, and you're going to have more questions. But, of course, we were really verbalizing all the things that we have going on as we go through it. you're going to be doing your internal monologue thing and probably talking to yourself a little bit faster. Section 2 is not really worth so much for us to go through, and we're not going to go through it like we just did. And the reason why is because in this exam, you will find, right after the answers, you will find, to part 1, you'll find rubrics for part 2. Those rubrics tell you what they're looking for in the questions. But let's talk about some strategies for answering these questions. The first strategy that I always tell my kids is to use the 10-minute period that you're given to plot your answer. And what I really want them to do at that point is to look at each question, to read it, and to use the information that they're given. 1 and 2 are worth 10 points each. In order to get a point on an essay, you need to basically make a factual statement. Like a factual statement is a point, right? Something that addresses what's being given to you. So just looking at the distribution of points in the first question, predict the distribution of flies in the chamber after 10 minutes and justify your prediction. It's probably 2 out of the 10 points right there. You've got that bold term for predicting the distribution, and you've got that bold term for justifying your prediction. I would predict my distribution for 1 sentence. I would justify my prediction in 1 sentence, maybe 2, and then I would move on. Propose 1 specific improvement to each of the following parts of the experimental design and explain how the modification will affect the experiment. So I'm going to propose, and then I'm going to explain. That's probably 2 points for each of those. So that's probably 4 points in the question. So I'm going to do what I need to do there, and then I'm going to move on. C, the experiment described above is repeated, and the data are shown in the table below. Perform a chi-square test on the data. Specify the null hypothesis that you're testing, and enter the values from your calculations in the table below. Just estimating this, probably doing the chi-square is probably good for a point. Probably specifying the null hypothesis is good for a point. And then putting in the values in the table is probably somewhere in there. Maybe that's 2 points, right? It seems like it might be. I don't think I'm going to get a point for doing the chi-square if I have the wrong values in the table, right? I think the way that I'm going to do the chi-square and show that I get that point is by doing it correctly. Explain whether your hypothesis is supported by the chi-square test and justify your explanation. 2 points right there. And then briefly propose a model that describes how environmental cues affect the behavior of the flies in the choice chamber. Probably 1 point, maybe 2. So if we go back, what did I say? I said part A is 2 points. I said that part B is 4 points, so that takes me up to 6. I said part C is probably 2 points, so that takes me up to 8. I said part D is probably 2 points, so that takes me up to 10. But that doesn't mean that I stop there. I also need to do E, right? And that's probably good for a point or 2 points in there. I am going to write, as a student, more points than I actually think I can earn on the test, so that I have a little bit of a buffer in there, which positions me well to get the maximum number of points I can on the question. The other thing is that the way that the rubrics are set up, as I'm sure you know, you cannot get to 10 points if you do not address all parts of the question and get at least 1 point in each part of the question. And in some, depending on the way it's set up, you might need to get more than 1 point in a particular section in order to wind up with 10 points at the end. But you don't want to waste time. And so using that 10-minute period to briefly sketch out sort of how many points you need to get in each section and use that framework of points for how many statements you're going to make in each section, that's incredibly important for writing an efficient essay. Anything that you're putting down on the page that isn't about the prompt is not going to get you any points. Those tips were brought to you by the one and only Dana Kopatich, Copat Scholars in Menominee Falls High School. You all work really hard, and she is going to truly miss you next year.
Oh.
Bob, anything you want to add to question 1?
Maybe not to question 1, but I also advocate to my students to use a little bit of time at the beginning to go through the free response questions and rank them.
Good idea.
Hardest to easiest, or vice versa. I think you and I are the same mind about this. This is really the only time of the year we ever talk about points, and this
Correct.
is all about getting your points. And so if you have those easy ones that you can do, you might want to start there. I think a lot of kids go in order, and sometimes that's a mistake because of time. If you go through and see which ones you know and you can get points on, do those, and then come back to the ones that might take a little bit more time. That's something that I encourage kids to do. I don't know if it's the best strategy, but it's a strategy that I encourage them to do for time management. So.
Nice. I don't think we're going to go through the rest of these questions here tonight, but what you can do is some of the following. The first thing that you can do is you can, after this, write those questions, or at least sketch them out, compare your answers to the rubrics, and see how they compare. And then if you have questions about it, you can always get in touch, probably primarily with your teacher, who probably wants to help you this weekend with some last-minute information. But you can, of course, always leave comments below the video, and you can get in touch with both myself and Mr. Kuhn very easily. You can search our names on Twitter, and you'll get right to where we are, or just on the internet more broadly, and I'm sure you'll get to us really quickly. It doesn't seem like a good use of our time tonight to really go through the rest of these questions, because you have those rubrics in front of you, and using the strategies that we just discussed, I think you'll find that you're in a pretty good place in order to address these questions as fully as possible. But as we close out the evening, we have a couple of final shout-outs that we need to give. Bob, do you want to give the next one?
Yeah, Kristen at Marcellus BioBrains, Marcellus Senior High School. Thanks for sticking it out this year. You've taught me a lot, and I hope you've learned some things and had some fun along the way. Go Mustangs, you got this.
Nice. Kate Fisher, Miss Fisher's AP Bio class over in Oregon City High School, which I assume is in West Virginia, says you can. You will. You can do it.
That's like a really Yoda moment there. Matt Bearcout, Beachy Bio. Now, I'm assuming this is I in my mind, okay, it's New York.
Yeah, of course it is.
Oh, I was envisioning it was California.
Oh.
No.
They probably wish they were.
Where the arts and sciences are one because you don't understand what you can't see.
Nice. And, of course, the inimitable Miss Melissa Green over at Deer Park High School in Deer Park, New York, would like to wish her Period 1 and Period 6 AP Bio classes all the luck in the world. They're awesome, and she can't wait for you guys to rock it on the test. Because, as you guys know, arguably the nicest biology teacher in the entire country.
Was she there when you were there, or did she take your class?
She has both been there when I was there, and now that I am no longer there, right? She now runs the entire show. But wow. You know she's nice, Bob, because she had to put up with me for 13 years. So that wraps it up for our little AP review session. We hope it was useful. We really are not just trying to ditch you guys on the part 2, but we've gone on for quite some time here. And really the best thing you can do at this point is write those essays, look at those rubrics, and get the clarification you need, either from your teachers or from us. But certainly, if you catch this either now or at some point in future years, right, or if you're, aliens descend to the planet and want to take the AP Biology exam several thousand years from now and watch this archaic video, we wish you all the best of luck on the exam. You guys will do great.
Yeah, good luck.
All right.